Chelsea Specs Lab, and today I'll be talking to you about incorporating developmental genetic information into trait evolution. And so I'd just like to start off my talk by discussing the hierarchical nature of biology and the different processes that act on different planes. So here we have a simple a drawing I like that kind of discusses the hierarchical nature from genotype to phenotype. And so the type of processes that we have is down here at the genetic level, we might have mutation. These mutations might shift some developmental parameter into a different parameter space, like changing gene expression. These gene expressions might then be manifested into some adult phenotype, which in here selection sees and filters through, and that produces the phenotypic variation that we see in nature. And so we as evolutionary biologists have models that account for these different processes. So for example, up here at the top, we have these models, these continuous time Markov models for discrete evolution and Brownian motion for continuous evolution, which don't need to be applied to adult phenotypes, but often are. Down at the bottom, we have models of nucleotide substitution that account for mutation, or thinking of it a different way, it's coalescing back in time using the coalescence. And we have models that kind of attempt to bridge the gap between multiple hierarchical planes, like these state-dependent speciation extinction models. But I argue that we don't have the strongest models that account for explicitly account for developmental parameters. There are models that exist in the literature that are not evolutionary, like these morphospace models, and today I'll be talking about that. And specifically, the system that I use are inflorescences, which are branches of flowers. And so here we have some diversity panel of inflorescences. And so by nature, these are continuous traits. They vary in the number of flowers, branch lengths, the leaf position, but oftentimes botanists classify these into discrete states defined by boundaries. And in particular, I will be talking about umbels today, which is these on the far right, and in specific the Amaryllidaceae umbel. And Amaryllidaceae is a family of plants that includes things like onions and garlics and amaryllis. And this inflorescence type is where all the flowers originate from a single point that you can see right here. And the reason for this is because there's a really nice botanical literature of developmental morphology on this. So if you were to take an onion and cut it in half, and we're lucky enough to find an inflorescence meristem, or the inflorescence stem cells, and you were to study the flower arrangement of these young flowers, you see this very interesting pattern which resembles a yin kind of on one side and a yang on the other side. And here's a little schematic of that. And early botanists conjectured that the way that you'd arrive at this highly condensed structure is by coming from the cyme-derived ancestor that had a branch rearrangement followed by differential branch elongation. And so here we have a hypothesis of topological change that's specific to this family. However, umbels are a convergent trait in plants, and so we see them in other families like the carrot family. And here it is thought that they arise from a raceme-derived ancestor that just had differential branch elongation. And so as evolutionary biologists, we see hypotheses of topological change that have yet to be studied in the phylogenetic framework. So we can simply ask this biologically motivating question: what is the evolutionary origin of the Amaryllidaceae inflorescence? And one way we can get at this is by making an inference of the ancestral state of this condition to see whether it is cyme-derived or raceme-derived. And so to do that, we need a phylogeny, we need data for the tips of the character states, and we need a model of morphological evolution, or a model of trait evolution. And so briefly, I'm not going to go into the details, but feel free to ask me afterwards. I inferred phylogeny using a supermatrix approach. I mined GenBank using this program SUMAC, and I inferred the phylogeny and did divergence time estimation in red phase using some fossils. And this is what we got, yay. And so for character coding, me and my undergrad Jason went through the literature and collected characters of discrete of the inflorescence states. And here we have them displayed out on the phylogeny a little bit. And now we'll be talking about these models. Well, the first thing we found was that we didn't have this intermediate form in this first hypothesis. And additionally, we had an additional state, this panicel, that we didn't see, that are not explicitly stated in these hypotheses. And so this simplifies our model. And so essentially what I did is I set up an ordered MK model that either allows for cyme-derived umbelness, so by the single error, or raceme-derived umbelness from this error over here. And since the models don't specify any, this is in the likelihood framework, sorry, since the models don't specify any anything about the additional transitions between the other states, I kind of did a pairwise comparison from equal rates to R rates difference, and I performed model selection. And so here we have the results of the model selection, and in bracketed the 4 best fit models with a delta AIC score of under 6. The next one up is 21. And these 4 models, 3 of the 4 are all raceme derived, so only allowing racemes to come from umbels, and except for an ART. And as you can imagine, if you infer ancestral states under these conditions and look at the node of interest, we see that it is raceme derived. The ancestor of umbels seems to be raceme derived. So this was a little bit unbelievable, I'm sorry. And so just to take a sanity check, we wanted to make sure that our model was able to it was an adequate representation of our data. So we did model adequacy following a Penn et al. 2015, which essentially we simulated characters using our best fit model, and then we did a t-test to see whether our observed data was statistically similar to our simulated data. And so here is one of the test statistics that we used, which was frequency of the state at the root for the different states, and they all fell within the bounds. The other 2 test statistics that we used were parsimony scores and the tip distribution, the state distributions at the tip. And so we can preliminarily conclude that the MRDC inflorescence is raceme derived. And as I said, this is a little bit, depending on how you think of evolution, and because it's not including developmental data, it's a little bit, wondering if this is a little bit actually unbelievable, if perhaps because we're not accounting for this developmental information, that we're misspecifying our model of trait evolution. And so one way, kind of going back to this biological hierarchy, we're kind of operating on this top plane when we really should be including information from here. And one approach that I think that we can use to do this is through these morphospaces. And so here we have this transient model. It's not an evolutionary model, but it's a model of branching architecture in plants that works like this. Essentially, it models this vegetative factor here on this graph. And this vegetative factor is a function of time that slowly decreases. And as this decreases, once it reaches a certain threshold, it'll generate a flower. So these lines here, this blue line, corresponds to this blue branch, this yellow line to this yellow branch, and this red line to this red branch. So whenever you have a branching event, and the branching event is set up by a Lindmeyer system, you get a spike in vegetativeness. And depending on the function of the rate of decay of this vegetative factor, you can produce different inflorescence architectures. And so if you were to cut across the morphospace in this direction, which is from left to right or up and down, you can generate these diverse structures which match up to the inflorescence architectures that we see in our trait. So now we can define a trait not just by a discrete character, but by the bounds of this continuous character defined by this vegetativeness. And so one approach that we can use is we can use this threshold model, which models a discrete, which assumes that discrete traits are underlined by a continuous variable, and this continuous variable is usually modeled by the Brownian motion. And it's called the liability. So on this figure from Revell 2014, you can see that this liability is being evolved, and once it passes a certain threshold, the state changes from blue to green or blue to red. And what's really nice about this model is that it accounts for some aspect of memory. So if you go off too far into one direction, it's more difficult to get back to that, which is more realistic. And if you look at our morphospace, this morphospace defines the bounds of this theoretical threshold or this liability. So what we can do is we can, what I did is in RevBase, I set uniform prior distributions for my observed data at the tips that are defined by the boundaries of this threshold, and I essentially modeled Brownian motion under it. And although this model is more realistic, unfortunately, it seemed to have too much uncertainty. So here we have the phylogeny and the tip of interest, the node of interest rather, and here we have the posterior estimate of this vegetative factor and the 95% confidence interval along the back. And as you can see, that this confidence interval spans the 3 states on this morphospace. So there's too much uncertainty to accurately state what state there is. But in the future, as you guys may have noticed, that this morphospace is not defined by a single character, it's actually multivariate. And now there's multivariate approaches to account for uncertainty. So that's one aspect that I'm looking into. And additionally, this model While it's a developmental genetic model that, sorry, I forgot to mention that this vegetative factor actually reflects the gene expression in Arabidopsis. There are other models that are rather just morphological, so they just generate virtual plants based off branching architecture and branch length. And we can fit these different morphospaces to data, I think in a Bayesian approach, and do model selection by Bayes family. And so, in conclusion, continuous time Markov models were able to infer the ancestral state to be received rather than signed, and they were adequately able to simulate data that is similar to our observed data, and incorporating developmental genetic data using morphospaces perhaps provided a more realistic model of trait evolution, but there was too much uncertainty at the node. And with that, I'd like to acknowledge my lab, the Spec Lab, and specifically two members, Carrie Tribble and Joyce Cherry, which are jointly advised with Carl Ruffles, and specifically Will Freeman, who helped me with the web-based stuff, as well as my funding. And I will take any questions. Yes.
It's just a small comment that too much uncertainty might just reflect the degree of uncertainty you actually have about the state. And so I wouldn't use that as a criterion to decide whether the method is a good one or not, because that actually might reflect the genuine uncertainty that you have about.
Sorry, that's a good point. Yeah, I guess right. I meant.
It's not too much. It might be the right amount.
Right. Oh, that's true. Right. Maybe the phrasing is for this data. It might not. This method
doesn't give you the answer, but that might be. The answer.
I think
it's not knowable.
Right.
But I do think that idea of doing multidimensional threshold is certainly pretty intriguing and totally possible.
Yeah, for sure. Yeah, I think there's other. I have these cool. You guys may all be familiar with the famous snail shell data, morphological data set for this cool color pigmentation of snail shells, which they actually did some comparative methods on. But I think thinking about your phenotype and thinking about where your phenotype comes from would be really powerful using this model. Thank you. All right. Well, thanks. No one has any questions? I'll be around, so let me know if anything comes up.